This is a production of Cornell University.
Hi, it's been great being here so far. It's a lot of fun being in a plant biology department. I've been in earth science departments my whole career, and it's been very nice talking to so many actual botanists. But even though there's a lot of plant biology in this room, I think it's important to explain why I'm a plant biologist and what I think of my plants, because it very well may be different from the way a lot of people think about plants. For me, the real draw for plants primarily is the fact that they have cell walls. And an awful lot falls out of that as far as what you can do with them as fossils. This is a fossil that's 300 million years old. The snails are just shells. The plant in between is a plant. We have all of this anatomy. It's all there. And given that plants are largely physical creatures involved in conducting water and so on, we can actually say a lot about how this thing functioned based upon having that anatomy present. The original cell walls are preserved. That biochemistry is there, so we can talk about the evolution of different biochemical cell wall compounds and so on. And also, kind of perhaps less immediately, but also very importantly, because they have cell walls, there's no cell migration. So their development is much simpler than what you'd expect to see in an animal with gastrulation and so on. And you can look at the final adult morphology and say something about how it got there in a much more direct way than you could with an animal and so on. On top of that, plants are all trying to make their living in the same way. And nearly all aspects of land-plant form has evolved multiple times convergently based upon that. So we have lots of independent experiments looking at the evolution of different structures. So if we look at this phylogeny of living vascular plants here, the lineages make it back to the Devonian and so on. It looks like, if you know what these guys are, it looks like there's a lot of morphological continuity there as far as roots and leaves and so on. But the common ancestors of these lineages are very small and simple. And most of what it looks like these things might share has actually evolved independently. And that's something we can directly assess from the fossil record, where all these guys have leaves that look kind of ferny, but we look at slightly older members of those lineages, and you're going to see nothing but these things. Slightly older, there won't be leaves at all. So we can actually trace the evolution of these things and compare multiple evolutions of very similar structures. And finally, something that it's taken me many years to really fully appreciate, that animals are cute, right? Or they're scary, whatever. But plants, they're the carbon cycle. They're all the biomass. They are the hydrologic cycle and all the water in the atmosphere. Plants are climate. And so the evolution of these guys and the evolution of their physiology really is the evolution of the Earth's surface in a very direct way. And there's been a whole lot of turnover through time in what those plants are like, and that turnover matters, right? There's a lot of focus on this kind of turnover down there with the animals, but they're really kind of passive recipients of a world that these guys are making for them. Now, my heart of hearts lies down here in the Paleozoic and all the crazy beasts that are down there. Today, I'm going to focus on these guys, the flowering plants. Now, I am not an angiosperm person. They scare me. There are far, far, far too many of them. I'm not an angiosperm person. But I think that has some utility, too. Looking from the outside in towards the angiosperms can give you some perspective on what it is that they're doing differently that it's harder to see from the inside. And then hopefully, if I have enough time, understanding what's going on here also reflects back on what's going on back there. Okay, so my primary kind of entry into the fossil record has always been leaves. Initially, I was interested in leaf development and how we can use venation patterns to say something about where growth is occurring within these leaf structures, how laminate surfaces evolve multiple times, and so on. I was doing that from in my thesis and so on since then. And we can use venation patterns to say something about sequential appearances of morphology, indicating sequential appearances of different developmental modes showing up in multiple independent lineages here. Over time, I got more interested in the fact that these things are not just passive records of development. They also matter for how this thing is functioning physiologically. They're conducting the water, and water matters if you're a plant. So I got more interested in the physiology, and as I did, I was amused by this a basic little lie in the way I've always presented this. Right? This might as well have been from my thesis defense here, indicating about how ferns can very closely converge upon venation patterns seen in angiosperms. But there's a lie in the way I presented that, which is that they're not presented at the same scale. If they were, they would look like that. There's a lot more veins in the flowering plant. And this actually is going to end up mattering quite a bit for how these things can function. So if you're a plant, you're trying to do photosynthesis. You're trying to take in carbon dioxide from the atmosphere, and you cannot help but lose water in that process. So in order to keep on taking in carbon dioxide through your stomata, you need to be able to replace the water you're losing. And if you cannot maintain that steady state, you have to close the stomata and you have to stop photosynthesis. So the ability to move water very much matters for photosynthesis. And hydraulic capacity is going to be very strongly correlated with photosynthetic capacity. And hydraulic capacity of the whole plant is very much tied up with the leaf. If we look at the resistance of the entire pathway of water through the plant, at least half of it, sometimes 2/3 or more, is tied up in the last little bit that is represented by the leaf. You go up through the roots, up the stem, out the branch, to the leaf, those last little bit. And even within those centimeters, the last couple centimeters, the reason there's so much resistance in the leaf is actually tied to the last couple microns, right, the last millimeter or so, which is the fact that throughout that hydraulic pathway, the water is moving through pipes designed for water transport. And it's when they leave those pipes, this is a cross-section of a leaf here, when you leave the vasculature here to make it to the stomata, you're moving through tissue that is not designed for hydraulic transport. And much of the resistance as a result is tied up in that pathway through the mesophyll. So the hydraulic conductance of the entire plant is very much tied to the leaf. And within the leaf, it's really tied to this path length from vein to stomata. So anything you can do to change that path length is going to affect the hydraulic capacity of the entire plant. And the easiest way to change that path length is just to change the vein density. If you have more veins, those little blue lines are going to be shorter. The maximum distance water has to travel from the vein to the stomata is going to be shorter. And to demonstrate that this really does matter here, this is looking at the distance from vein to stomata. And the shorter that distance is, the higher the leaf conductance to water. And the higher the leaf conductance, the higher the photosynthetic rate that that plant can do. Right? So this is all very much tied to these vein characteristics. So this is looking at distance from vein to stomata. If you flip that around, you get vein density, which is a much easier term to work with. And that's just looking at the millimeters of vein per square millimeter of leaf surface there. And so if you look at that here, this is vein density versus maximum transpiration rate. And yes, there's a very good relationship here. And now the fun thing is that this is something that is readily measurable in fossils. We can work with this trait. And that becomes very, very important. Right? So here's a living cycad. Living cycads are very slow. They grow very slowly. But you would never impugn a Triassic cycad and say that, well, they have to be growing slowly like the living cycads. For all we know, these guys have been displaced by the spread of angiosperms. Maybe they were more ecologically diverse in the distant past before these transitions occurred. But no, I can show, I've looked at a variety of fossil cycads. They all look like the living cycads. They're going to be stuck way down on this spectrum here. So it becomes a very powerful tool for looking at the physiology of fossil plants and physiology through time. And we looked at a whole bunch of different plants. And first off, don't worry about the colors. That had to do with a specific argument about leaf development and how that might be tied or not tied to potential vein densities. The pattern to look at here is the fact that these guys down here, which are the flowering plants, go way farther out on this axis than anything else that has ever lived. And this includes a lot of exclusively extinct lineages of seed plants and so on, as well as ferns up here as well. Now, perhaps the best way to look at this is this graph here, which is vein density through time. Vein density is incredibly stable in all non-flowering plants. It's around 2. It can go down to about 1 millimeter of vein per square millimeter of leaf surface. It can go up to around 5, but that's about it. Until you get the evolution of flowering plants, where the average is about 8 to 10, and it can go up to 25. The lower bound is still the lower bound, but the mean and the maximum is about 4 times larger than any other group of plants that has ever lived. And you can see what some of these guys look like in practice here. So this is a fairly typical flowering plant. That's a fairly typical fern. You can't even see the smallest veins here, whereas you're seeing everything in that fern. I had to add this in. I gave a talk once, and there was a molecular biologist who even worked with leaves, but he had seen these pictures of ferns that I was putting up, and he assumed that there was more orders of veins that he couldn't see that were there, and that I was just showing the bigger veins. No, those are all the veins. There's nothing else there. And that really is going to end up mattering physiologically. I would like to pause, though, and say that the veins are not magic. Right? This is not a magic leaf. If I took one of these and stapled it to a cycad, you would not get a magic fast-growing cycad. You would get a dead leaf. And that what this is doing is integrating a lot of the biology, the whole biology of the organism. It's just in a way that we can actually then measure in fossils. To get an illustration of that kind of integration, I'm going to do a bit of a tangent that will actually come back and be important later on, where there's a paper that I was not involved in, but the senior author here is Maciej Zieniski, who's a close colleague of mine. And what they did is they made artificial leaves out of gel. And they carved little channels in them, ran a tube to a balance, and they blew air over the surface and evaporated water from this surface as an artificial leaf. And what they were able to demonstrate is that as you add more channels, more veins, the amount of water you lose increases, but then it rapidly saturates. And where that saturation occurs is going to reflect the thickness. And what they found is that optimal placement of these veins, if you're trying to lose water, is when the distance between the veins is the same as the distance from that vein to the evaporative surface. If you have more veins than that, it doesn't matter, because you're just going to flatline. And so this leads to expectations of what leaves should look like in cross-section. These are all cartoons, are all cross-sections of leaves. These are good leaves. These are what leaves should look like. If you have a lot of veins and they're close together, this should be in a thin leaf, right? So that D and the delta are the same. If you have a thick leaf, you can have two different systems going on. If these are medially placed, then you should have a very low vein density. But you could also have these things offset, because what matters is not the actual thickness of the leaf, it's the distance from the veins to the evaporative surface with the stomata. These are good leaves. These are bad leaves that should not exist. There is no point to having a lot of veins if they're far from that evaporative surface, because that evaporative surface can't see them, and it's just kind of wasteful, the construction and the displacement of photosynthetic tissue and so on. Similarly, this should be a problem, because the tissue immediately in between these two veins should dry out. It shouldn't be getting enough water at all. So you should actually be kind of actively killing stuff down in there. So we have expectations of what leaves should look like. And then they went out and they measured a bunch of plants, and wow, that's a biological paper, and they get a relationship that good. So this is very impressive. This is the one-to-one line that they wanted, and everything is right there on the one-to-one line. From thin leaves down here to thick leaves up there. When I saw this, when this came out, this was when I was first kind of sitting down and measuring vein density a lot. And that's like all I was doing with my time for a while there. And this paper immediately bothered me, because there was a big problem with this. And what that problem was where are the ferns? Ferns have thin leaves and they have very low vein densities, so they should be plotting out here, not on that line. I know Machi very well. He's a very good scientist. I don't think he screwed up. But I had a theory about what was going on, which was that the authors were all either hydraulic physiologists or actual physicists. They were probably just went out and they grabbed a bunch of plants. They're on a university campus. They just grabbed stuff and they measured it. If you do that, I mean, you're sampling this kind of environment. And what I suspected was going on was that it was not just a simple line, that there was a slightly more complicated relationship for these different types of leaf types. These are the good leaves here on the line. These are the bad leaves I mentioned. This bad leaf should just never happen. Over-investing in veins, there's no reason to ever do that. So that shouldn't exist. But this could exist as long as you live in a sheltered environment. So what I suspected went on in this original paper and how they got such a good relationship is they were only sampling out here as far as degree of exposure. And all those things are going to be one-to-one. But if you got things living in the shade and wet environments and so on, they could fall off that line, and that's where you're going to find the ferns. So we went and we sampled a whole bunch of stuff, and yeah, that's what it looks like. It's not a line, it's a triangle. Now, the first thing to say about this is that is a really glorious triangle, right? They really did get the hydraulics right. And I had nothing to do with that. I wasn't on that first paper. But they really did get it right, because there really is nothing below that line. The one-to-one line really is what matters for this. So I think that's a very good indication that they really did do the hydraulics right. They understand what's going on. But yeah, we do fill in a triangle here. Knowing what all those different symbols are, there really is an ecological gradient there, where the things in exposed environments are on the line, and the things that fall off the line are going to be in much more sheltered environments. And there's even gradients within the ferns and stuff like that. So we saw what we expected to see. But we also saw something that I didn't really anticipate, which is there's a big kind of separation of different phylogenetic lineages as well. So I already mentioned the ferns. And yes, there's an ecological gradient in here where the ferns down there are tree ferns in exposed environments and so on, versus guys that are in deeper shade. And they all have thin leaves. So none of them can be very good, although there can be better or worse. Gymnosperms also have low vein densities, but they can have thicker leaves. So they can end up actually kind of grading onto this line by virtue of having very thick leaves. And again, there is also this ecological gradient. And I'm not showing the data, but all the fossils we looked at are actually consistent with that. suggesting that the ecologies that we're seeing in these living guys are consistent, and ferns have always been ferns, and cycads have always been cycads ecologically as far as what they can do. So we set up this system where the canopy and smaller trees in more open environments are going to have these thicker leaves and these various seed plant lineages that are capable of doing that, whereas these guys are going to be more restricted to these sheltered environments. But then all the angiosperms we looked at violated that in a very interesting way. It doesn't matter where you are ecologically, the angiosperms are on the line. Deep shade angiosperms are also on the line, even though they don't have to be, which is very interesting to think about their evolution. Except that's derived angiosperms. Here, the red angiosperms, they look like the ferns. So across the basal angiosperm phylogeny, they start out all looking kind of very ferny in these relationships, and then once they get on that line and more derived angiosperms, they stay there forever. And you can see that here. This is plotting it. This is just looking at the ratio. So you don't know where you are on this line, but you know how far off the line you are. And this is everybody here. The ferns are up here. They're not so hot. The various gymnosperms are slightly better, but not so much better. And then once all this is the angiosperms, Anita, basal guys, this whole backbone is poorly optimized. But the core eudicots and the derived monocots, and specifically the Lorales, but the Magnoliales and Lorales are better than the Piperales and so on. And so the backbone of this would have been very poorly optimized, and there's several independent lineages within the flowering plants that figured this out. So there had been kind of this stable system through time. The things that can tolerate being in open exposed environments versus the things that couldn't, but could handle this more sheltered circumstances. And then the angiosperms came along and completely violated that. Whereas these things would have been, their thin leaves would have been capable of a lot of gas exchange, but didn't necessarily have a lot of need for that gas exchange. That's why they could handle having so few veins. Whereas these guys are much thicker and are therefore capable of much less gas exchange. But they're the ones that actually had the light and so on. What angiosperms did was they figured out how to take leaves that should have been in the shade and put them at the top of the canopy. And that's something that basically only they have figured out, which speaks to a lot of what they're doing and how different it is biologically. And these aren't magic. These really are integrating an awful lot about the overall biology of the plant. Okay, these guys are different. For the rest of this talk, I'm going to explore what that actually means for the Earth and for the plants and the ecosystems and so on. To start out, I'm going to talk about this climate here. So this is a graph I already showed you, how vein density scales to maximum transpiration rates. Transpiration matters. Particularly if you go to tropical systems, recycling of rainfall can account for 30 to 50% of the rainfall that falls on a rainforest. It falls out of the sky, the plants suck it up, put it back up in the sky so it can fall down again. This is showing this kind of cloud seeding over the forest in the Amazon, and it's present over the forest, not over the ocean in the same way. So that matters. And I'm saying that those transpiration capacities up here would not have been present before the evolution of flowering plants in the same way. So that the evolution of flowering plants is relevant to thinking about the evolution of tropical rainforests. Now, I'm not saying that flowering plants invented rainforests. There will always be a tropical rainy belt, right? Here it is stretching across the ocean. That is not the distribution of seagrasses. This always would have existed. And similarly, if you have mountains near a coastline, they can squeeze water out of the sky. So there always would have been little distributions of rainforests. In fact, angiosperms had to have evolved in them based upon the ecology of basal extant lineages. So they did have to pre-exist. However, transpiration is important to these systems. They may very well have been important in kind of the spread of these things, depending upon how important that is. And the question becomes, well, how important is it? So I engaged with a postdoc, Jung-Yoon Lee, who is a climate modeler, and we looked at some of these things. Plants are part of climate models as they exist, and they have physiological parameters in there. And what we ended up doing is we just turned the one knob that had to do with these physiological rates and looked at what the outcome was. And these are standard models for atmosphere and ocean. Using modern circumstances, modern sea surface temperatures, all we changed was the transpiration capacity of the plants. And it mattered. This is looking at the Amazon basin here. This is a difference map. So it's showing how much rainfall is lost in the absence of flowering plants. And we have the same biomass of plants. We just changed the physiology of the plants to a non-angiosperm kind of mean average value. And at the center of that bullseye, we're losing about 1,500 millimeters of rainfall per year, which is a lot. If we look at that temporally rather than spatially, but this is still over the Amazon basin, there's a variety of interesting things to see. So the blue is present day, and the blue versus the black shows how closely the model corresponds to actual values. And in the absence of the flowering plants, the basin is hotter and drier, and it's much more seasonal. Seasonally, it's up to 5 degrees hotter, because at the tail end of that dry season, transpiration becomes very important for the latent sensible heat. Becomes very important for cooling off the forest, and it gets much, much hotter once you turn off that transpiration. So we're hotter, we're drier, and we're more seasonal in the absence of flowering plants being present in the system. And overall, the area of what gets defined as ever-wet rainforest, based upon number of rainy days per year, drops by 80%. There's still a rainforest band, but it is much more confined to where the ITCZ, that tropical rainy belt, would be situated. It's not that massively expansive Amazon basin that we have. Now, I'm showing you the Amazon, because that's where it works the best. You can see that here. There are global effects. They are less severe in other places, and they're less severe for fairly obvious reasons. Africa is high topographically, and you have the East African Plateau, which constrains where you can get precipitation from. So it's hard to make Africa wet. And Southeast Asia is a bunch of mountainous islands in the tropics. It's hard to make them dry. You can't make them dry. So there's reasons for why the impact is less in other places. But like in Eastern North America, it's still a 50% reduction in precipitation. It's just a different color because there's less overall. It's still 50% of what's going on. And there's reason to think that what's going on in the Amazon basin, I think, is perhaps relevant to some things that are going on in Earth history. This is the Eocene here. The dark hatching is a distribution of tropical aspect rainforest, even though some of these things are at rather high latitudes. I think the fact that there was this kind of extreme spread under these warm circumstances in the Eocene may very well have been related to the transpiration capacities of that vegetation, which was flowering plant dominated. And one way to think about that is if we, this was not the only time the Earth has been very warm. If we go back a little bit earlier, oops, that's the wrong way. Back to earlier in the Cretaceous, it was also warm. But what we're talking about in North America and so on is seasonal aridity. And the difference is turnover in what that vegetation was composed of. What those forest canopies would have been created of. There would have been much less transpiration in this world. In any case, if you just thought about the physical nature of this world, this should have been the wetter world. Because this is when we had lots of internal seaways breaking up these continents, and the pathway for moisture movement should have been much less. So if just kind of based upon those reasons, this should be wet and this should be dry, but that's the opposite of what we actually see. So I think this could have been very important earlier in Earth history, earlier in the history of angiosperms in particular. And it also sets up a very interesting dynamic because plant diversity itself is tied to precipitation levels. This is some work of Kreft and Yetz here, where what they're showing is that this is potential evapotranspiration. So this is kind of how warm it is, how much water you could evaporate, which is related to solar influx. And what it shows is that looking at diversity in lots of localities around the world. And what it shows is that if you're cold, getting warmer matters, right? That's this area here. But once you're relatively warm, getting warmer still doesn't make a difference. What does make a difference is precipitation. And this is also where angiosperms are the most hyperdiverse. So they are creating the very environments where much of their diversity is situated. They're propagating them out much larger than they otherwise would have been based just on physical parameters. And then that does matter also for animal diversity and everything else. This is looking at primate diversity, and animal diversity is also correlated with rainfall abundance, evenness, and rainforest area. Do the monkeys care that it's raining? I don't think the monkeys care, but the plants care, and they care about the plants. So terrestrial diversity as a whole is going to be very much impacted by the evolution of this physiology that is unique to the flowering plants. And I think you can perhaps even see this in other lineages of plants. This is a fern phylogeny where the green is evolution of epiphytic ferns. The lines there, you can't read that at all, but the lines here represent the KT boundary, which is the end of the Mesozoic, start of the Cenozoic. Flowering plants evolved around in here. And all these lineages of epiphytic plants are evolving after this transition to a flowering plant-dominated world. These guys are the filmy ferns. They're kind of crazy, but I think there's plenty of good reasons to ignore them. But if we look earlier in Earth history, where are the epiphytes? So this is the Triassic of Greenland. It would have been the warmest, consistently wet place at the time. The tropics at that time were more seasonally dry. So this is as close as you're going to get to a rainforest type situation. Plenty of these plants have been reconstructed as lianas or vines, but there's not epiphytes. And I think this may reflect this transition where epiphytes are going to be very dependent upon the regularity of rainfall. And that regularity is going to be buffered by the existence of flowering plants in these later time periods. So I think their evolution has greatly impacted climate in a way that then also impacts just diversity of both the plants themselves and everybody else that goes along with them in these ecosystems. That's climate. This is also, I think, going to be very relevant for productivity. Now, I showed graphs that were just very kind of clear-cut. Productivity should scale with vein density. It gets a lot more complicated than that, and bear with me, please, because productivity is also going to be dependent upon other factors, the most obvious of which is going to be atmospheric CO2. And that has fluctuated quite a bit. We're actually now in a very low level of CO2 versus the distant past. That initial paper on vein density, there was 4 of us. We were all expatriates of Missy Holbrook's lab. We were all postdocs at the same time there. And 2 of those authors were Tim Broadrib and Taylor Field. And they went off and measured a whole bunch of more angiosperms, more than I ever cared to do, because they're the hardest things to measure. And which was great. But then they also kind of developed a model over how this relates to carbon dioxide changes through time that I felt was very problematic. And what they argued was that the effective vein density is going to saturate at high carbon dioxide concentrations so that there's no advantage for high vein density when CO2 is high. And it's only as CO2 declined from the Cretaceous to the present that suddenly becomes these high vein density values become useful, and that's when these things are occurring. So they link the evolution of angiosperms to declines in CO2 through time. And they talked about this in terms of a big increase in photosynthetic potential. I was kind of concerned by this model, though, because the back end of it is problematic. So what this graph here shows, and it was just kind of in the supplementals, was different crops versus tropical trees versus non-angiosperms and what they do in the modern world, and there's these big differences between them. But sure, everything saturates at high CO2, but they have it saturating way the heck up at here. So what this is saying is any crappy fern, if you get enough CO2, it would be much more productive than maize is and sunflowers are now. And that felt wrong. Right? And this actually fits into a larger debate. I mean, this is evolutionary biology, and so the debate happens at the rate of a paper every 5 years. But a variety of people have come out on either side of this, where there's a variety of people that are very kind of invested in carbon dioxide through time and wanting to make this as the driver of much of what's going on in evolution. This is the paper I was just talking about, Broadrib and Field, and then a variety of these others as well, a lot of them led by David Bierling and his students. So there's this focus on this extrinsic driver of CO2. In other cases, and not always aware of what they're arguing against, I certainly wasn't aware of it in some of the cases I was arguing against this, are people that have focused much more on the intrinsic biology of the plants. I think the best known of these would be Bond's great paper about the tortoise and the hare and the differences in the potential ecology of angiosperms versus, he was largely talking about conifers, but you can expand it beyond there. And so, the same CO2 changes are occurring, but there's just no interest in emphasizing that as far as what's driving the changes in the ecology through time. And I thought that this model might be a very interesting way to test some of this, because the model that they use is reasonable. It's the same photosynthetic models that have been used since the early 1980s. They've been very well vetted and so on. But they found a way to include vein density in it, which is another way to get at the fossil record. And they actually set up some very interesting tests here. They emphasize this flat line and why there's no advantage to high vein density when CO2 is high. But they didn't talk about, which I find much more interesting, is just how steep this is. Because no other plants besides the angiosperms ever made it up here. But any plant can do this within this pink range here. And if this is an extrinsic driver, it should be affecting all plants equally. That's what's inherent in this graph here and how they have the ferns getting so productive as well. But what that means is that if you're down at this lower range of what other plants do, any incremental increase in your vein density is going to make a huge difference in what you can do as a plant. In ways that should not matter for the overall hydraulics of the plant, but what it just means is that when CO2 is that high and photosynthetic rates can be that high, if you're not doing it all the time, you're going to get overgrown just like that. So there's a huge disadvantage to having very low vein densities. And the expectation would be that the upper bound, maybe that just can't change outside of the angiosperms, but the lower bound in everybody should increase at times of high carbon dioxide concentrations. You should lose the lower half of this pink range here. And it just doesn't happen. So this is one of Berner's models of this is CO2 through time, showing the time periods that we looked at, and we look at vein density at these time periods, and there's no change. There's no dependence upon the CO2, what that lower bound looks like. The medians in all these cases are basically down in this zone where there should be nothing at all, and it's not really impacted. And I'm not showing the data, but there's a variety of CO2 models, and if you try the different ones, it's all the same. The location of these bars will change up and down, but there's always stuff up here and there's always stuff down there, and there's no effect on the vein density. So this didn't work. So something's wrong with that model, and yet it's a good model, right? So we got curious about whether we could figure out, rather than just saying this is wrong and walking away, saying, well, what happened, and can we improve upon it? So this is the model that they used, the way they incorporated vein density into these photosynthetic models. So they got leaf conductance via the vein density as per those empirical graphs that Tim Brodrib did himself. So that's how they got the leaf conductance. And then flow into the leaf is going to be dependent upon the leaf conductance and the water potential gradient. And then the flow into the leaf has to balance the evaporation from the leaf, which is going to be related to the vapor pressure deficits and stomatal conductance. So then we can get stomatal conductance from the vein density. And then that can be plugged into a model of the physical limits of diffusion on CO2 getting into the leaf. And then that can be solved simultaneously with a different model of photosynthesis based upon the biochemistry. And you solve those simultaneously, and that's how you end up with these curves here. So is there a way that we can kind of fix what they did to actually get something that worked with the fossil record? The first correction we did was getting back to that thing I showed you about leaf thickness, because they used all leaves being the same thickness, when in actuality it should in some ways correlate with the vein density that they're showing. It was very gratifying. We did that correction, and so they had some empirical data that they used to correlate their graph, and our correction actually matches their data better. So that made us happy. But then when you look at the resulting curve, it doesn't really matter. You still have a big disadvantage to the lower end of the vein densities that any plants can do. And this is showing the rate gain in assimilation with vein density here. And you can see how much it matters that, if you can creep up out of this zone here as far as the overall photosynthetic gain that would be possible. So that didn't help. We tried a variety of other corrections. They didn't really play with oxygen, whereas that's going to affect photorespiration rate, and oxygen also has fluctuated through time. We played with stomatal conductivity, which should go up and down with carbon dioxide concentrations independently of what's going on with the veins. We tried all those things, and none of them matter. So none of these corrections matter. And then they only looked at discretely, like low versus high CO2. We looked at it continuously, where here the colors are the assimilation rates, but we looked continuously at all potential CO2 concentrations. And you can just see how the colors stack up here. It doesn't matter. Once you get out of this low CO2 regime, it matters immensely, getting a little bit higher of these vein densities, and they don't do it. So none of these corrections to what they did make any difference whatsoever. So we have to look a little bit beyond that. And the one solution that does work is if we just say maybe ferns can't be sunflowers. If we lower this VC max, the maximum carboxylation rate, and just assign different rates to different lineages, it actually solves everything. Because what ends up happening then is that you still have a plateau related to vein density and at different CO2 concentrations. But what happens is that at high CO2 concentrations, that plateau occurs right at the base of vein densities that plants do anyway. So basically, ferns don't care about CO2 concentrations in that regard. Higher CO2 concentrations, they're not going to be any more productive, is the argument there. And that it's really only the angiosperms that are potentially going up there. Now, that goes in the model as maximum carboxylation rate. But again, this is something where it's integrating the overall biology of the plant. Carboxylation in rubisco, that is very CO2 dependent, sure. And people have always kind of assumed that it should just track CO2. But you also have to, the whole rest of the Calvin cycle and the regeneration of RUBP, that is not CO2 dependent. And then there's a variety of other things that have to do with productivity, like phosphorus and nitrogen, that you need. If you're going to be that much more productive, you need more phosphorus and nitrogen. And there's some indication that that's actively kind of gets harder as CO2 concentrations increase. Or at the very least, it's unrelated. So there's plenty of reasons to think that you can't just amp up CO2 and expect everything to go along with it. Okay, is there other reasons to think that this might be true? If we take and plants, and these are all just numbers from the literature, and give them elevated CO2 concentrations, what happens? Yes, flowering plants end up here. Ferns and conifers that have been done don't. So this is consistent, right? I mean, there's no ferns out here potentially to go up there as far as their vein densities. And yet, you know, within the range that they do exist, there doesn't appear to be much of an impact of elevating that carbon dioxide concentration. People haven't focused upon that because they're more interested in these overall patterns and the big things up there. But there's no reason to think that these guys could have responded in that way based upon this. And it's even consistent in a very fun way with the fossil record. So this is data from a follow-up paper from these guys led by Taylor Field, where they measured a whole bunch of Cretaceous plants. The black circles are non-angiosperms, the white circles are angiosperms. And we look at the black circles, they say what I've been saying, that there's no response to whatever fluctuations of CO2 went on in this time period. They're just kind of merely continuing on with this flat line as far as their vein densities. Early flowering plants share that full range, including the lower half below that yellow line, which corresponds to that steep part of the gradient there. As they evolve the high vein densities and presumably the various physiological capacities that go along with that, they lose those low vein densities. So they are responding the way you might expect if this model was correct. But it's only the flowering plants that do it. So I think this is potentially a very useful way to actually bring the fossil record to bear on some of these relationships that have been proposed. These are, this is the graph I've shown explicitly versus CO2 concentrations. These papers, various papers from David Bierling's work, just is relative to time. And this is largely tied to CO2 fluctuations. And I think basically you can throw out the top half of all these graphs. They never got there. And I think there's even reasons to think, independent reasons, that they couldn't have ever gotten there, which is all of these graphs are ultimately dependent upon the work of Bob Berner and his modeling of CO2 concentrations through time. Berner is very explicit about what goes into his model. And plants are a big part of it because the primary drawdown of CO2 from the atmosphere is via silicate weathering. And that's largely mediated by plants. All the acids pumped down into the soil by roots and so on increases that. So the biggest feedback on CO2 concentrations are plants. And the idea is that if you have more CO2, there's more productivity, and that leads to the drawdown of more CO2, and so CO2 comes back down. And he tries, he has these sensitivity analyses looking at the levels of carbon dioxide fertilization. And it's right there in his papers of if you had as much CO2 fertilization as shows up in these graphs, then you wouldn't get any change in CO2 at all, because it would just be swamped out by the plants immediately sucking up that CO2 and it ending up with the silicate weathering. Ultimately, the silicate weathering of rocks on land ends up in carbonate rocks in the oceans. And so there's definitely a CO2 fertilization effect. But if it was as extreme as these papers rely, they wouldn't get the CO2 curve that they're using to generate those graphs in the first place. So it couldn't have been that productive. It just wouldn't have worked out in that way. So I think productivity really has greatly increased with the evolution of flowering plants as well. Otherwise, I mean, the baseline of these papers is that we're actually living in a world that is very depauperate and very less lush than it used to be, and I find that hard to believe on some level, right? So I mean, that's just an internal bias on my part, but it doesn't feel right to think that the most productive environments were ferns in the Jurassic relative to what we have now. So climate's changed a lot, productivity has changed a lot. And I think that as a last step, and this is just a couple slides, I promise, it's then interesting to use that to think back to the earlier record. And as I said, this is really what I like to think about the most. But this becomes very important because paleoecology has to be founded on modern ecology. And modern ecology is flowering plants. There's no place on Earth in the terrestrial environments where flowering plants don't represent at least 90% of the species. Even if you go to Siberia, most of the biomass will be the same couple species of conifers, but most of the species are going to still be herbaceous plants underneath that are all flowering plants. So modern ecology is flowering plant ecology. But everything I'm saying says that probably is not a good way of thinking about this system here, back in the Carboniferous. And there's a variety of implications of that. I'm just going to kind of touch on a couple. But this is kind of the stuff that interests me the most at this point going forward. This sometimes gets maligned by ecologists now. I don't know why. I kind of like it. It's useful. This is Grimes' triangle, looking at ecology based upon kind of different guilds of competitors, your stately oak trees and so on, versus weeds, things that reproduce very quickly, use a lot of resources, and then reproduce and die, versus stress tolerators. They're slow growing and also have a very low reproductive output. And this is related to intensity of stress and disturbance and so on. But two of these three vertices here are dependent upon high productivity. So basically, I think that this is angiosperms. And in a world without angiosperms, you still have some differentiation here, but we're talking about a much more limited triangle, basically. I mentioned this to Bill DeMichael, who's a paleoecologist, and he said, and his immediate response was, yeah, like I was on my high school football team. And if you know Bill DeMichael, he's about that big and not big in any dimension, right? But in a high school team, there's just less competition. Relative to the NFL. So there still can be differentiation within this space because it's much more limited than what we see in the modern world, based upon the fact that we're going to be much more constricted in this range of photosynthetic potential. And I think there's evidence that is going to be present. Because I'm talking about a world without weeds in a very real sense. And I think that may be correct, right? We have herbaceous plants, but we don't have annuals really in the fossil record. Now, in part, that's also going to be tied to angiosperm reproductive biology, right? So this isn't the whole story. But things like bracken is the one thing that gets often brought up in this kind of context as being a competitor. It's slow growing. It's not a competitor by any kind of modern sense. It's just that it's hard for once it's there, there's an incumbency effect and things can't grow up through it. But it is definitely not a weed, and it's not really a proper competitor by these modes. And kind of one last thing to think about in this regard, this is an oversampling of the Westphalian bee in the Carboniferous. So this is less than 1 million years. This is effectively one time slice. And I chose this time slice one because we have a lot of fossils from the Carboniferous, and also CO2 concentrations were about what they are now then. So even if you don't like what I said about carbon dioxide, photosynthesis would be tied to vein density then the way it is now because it's the same relationship. We have a lot of lineages here. I've sampled this about as well as we had in that original paper sampled modern plants. So we know this as well as any other time slice. And these are all from England, right? So this is all basically one flora. And there's some differentiation there. But it's all incredibly low relative to what's seen in living flowering plants. Everything is maximally what you would only expect to see in the deep shade if we're talking about an angiosperm system. And they're largely even lower than that kind of thing. Which I think is kind of fun to think about in terms of just the original evolution of vascular flora. It's often kind of couched as kind of reaching for the light as far as why we evolved trees on and so on like that. I don't think any of these things cared about light. I don't think any of them were light limited. They had other things to worry about. And that none of these plants were desperately wanting the light levels that a modern angiosperm tree wants. And yet that's kind of often when we look at, and this is a graph I put together myself many years ago, so I'm as guilty of it as anyone else. But very often when we think about kind of the Devonian evolution, the Paleozoic evolution of vascular plants, it's implicitly there that we're talking about ecological succession and what goes on in ecological succession. But these guys are all fighting over light in a way that I don't think was going on here. And it gets into questions of, well, why be a tree at all then? Right? If you're not trying to shade your neighbors and get to the light, why are you doing this? And I think that there can be a variety of kind of non-intuitive but interesting things to explore in this regard of it could in part be related to being long-lived, because plants keep growing and they can't really avoid it. It can even be involved with things like rooting depth instead, where if you're rhizomatous, you're never going to get roots that are that deep with these adventitious roots, where there can be a point to being a deep-rooted plant, but that's something that only trees can do. So it could be as much about what they're doing growing down as growing up. One example of this that's fairly local is Bill Stein over at Binghamton has a locality where they have these casts of tree stumps. And there's one kind of basal relative who would be one of these guys here, actually, although that actually might be a basal seed plant. But a relative of the seed plants growing rhizomatously. And it goes from about here all the way across that room. That would have been many, many years of growth. And throughout that growth, if the thing's going to stay alive, it has to keep growing. But if your rhizomatous growing horizontally, you're constantly changing your environment as you go. Whereas there might be a point to sitting in one spot if you're going to be long-lived and growing up. And it also allows you then to extract more nutrients at depth and more water and so on. So it might be more complicated than the easy analogy that's available from modern ecological succession and basing things on light. Okay, one conclusion slide. So flowering plants are different. And they're different in ways morphologically that translates to their physiology. And that physiology matters for climate, and climate is relevant to the fact that they're also hyperdiverse and biodiversity in these environments which they dominate, which is all environments. They're also more productive in a way that I think is not going to be related to carbon dioxide, the way it has been often related in the older literature, in the current literature. And the fact that they're so different, it makes it problematic to easily kind of translate from modern ecology to paleoecology and what's going on there. And it's kind of a last point here. I haven't talked about flowers. I've been talking about flowering plants. I haven't talked about flowers at all. And very often when people talk about flowering plant evolution, they're focusing on the differences in their reproductive biology. And they certainly are extreme. And they matter a lot in a lot of ways, both. But as far as their overall dominance of the planet, I think the vegetative biology is as relevant, right? Because I'm sure if you're going to have 250,000, 500,000 species, whatever it is, most of those have to be rare, right? And the reproductive biology of angiosperms is very good at keeping rare species. Sure, that's great. But they're also most of the biomass. And I think that really comes down to their vegetative biology. The reproductive biology allows them to subdivide all that real estate very finely, but the reason they have all that real estate in the first place, I think, is come much more down to their vegetative biology. And that's at least as important when thinking about the role of angiosperms in the modern world and how they evolved. Okay, thank you.
This has been a production of Cornell University, on the web at cornell.edu.